Welcome to the Canvector National Thrombosis Seminar Series. My name is Paul Geraz, and I'll be your moderator today. The Canvector National Thrombosis Seminar is a self-approved group learning activity as defined by the Maintenance of Certification Program of the Royal College of Physicians and Surgeons of Canada. This seminar series is brought together by the Planning Committee. And today's presenter is Dr. Ejafe Agbani. Just a brief background. Dr. Agbani received his Bachelor of Pharmacy from Obafemi Awolowo University in Nigeria. And this was followed by a Master of Science in Clinical Pharmacy from the University College of London. Subsequently, he undertook a PhD at the University of Strathclyde, focusing on cardiovascular cell biology and pharmacology. Afterwards, he undertook postdoctoral training, most notably in the laboratory of Alistair Poole at the University of Bristol, where he studied membrane ballooning and the procoagulant spreading of human platelets. Since 2018, he joined the University of Calgary, first as a research assistant professor. Subsequently, he's now a tenure-track assistant professor, and his laboratory focuses on thrombosis and hemostasis, procoagulant membrane dynamics, thromboinflammation, platelet function disorders, as well as cancer-associated thrombosis, and he utilizes advanced imaging techniques. He's funded either as PI or co-PI through NSERC, CIHR, Alberta Health Services, as well as the Canadian Hemophilia Society and British Heart Foundation. He's published nearly 50 publications in very good journals, excellent journals, I would say, including Blood, Circulation, as well as Journal of Thrombosis and Hemostasis. And the title of today's talk is on Platelet Procoagulant Membrane Dynamics Study, Mechanistic Insights, and Clinical Translation. There are learning objectives for today, and take away, Dr. Agbani.
Oh, Dr. Agbani, you just have to unmute yourself.
Oh, okay. Thank you, Paul, for the invitation. And thank you for the introduction also. So I have two here. I have two PowerPoints here. Which one is it?
You just have to share your screen, Dr. Agbani.
I can see two PowerPoints here.
You might have it open twice. I'm not sure. Just I can share as oh, there you go.
Okay. So during the course of this talk, I would talk about the dramatic morphological transformation that platelets go through during thrombosis and hemostasis, what we call PMD for short, procoagulant membrane dynamics. I'll talk about the functions and mediators of platelet procoagulant membrane dynamics. Then I'll provide some example of the translational value of analyzing PMD systematically. And then I'll finish up with a mention of a new innovation that we're trying to put together that is taking advantage of the systematic analysis of procoagulant membrane dynamics in platelets. And the first response of the human body to injury is really a vasoconstrictor reflex. Now, this is really an attempt of the body to bridge the gap and prevent the loss of blood. Now, this is immediately followed by what's called the primary formation of the primary hemostatic plug. Now, here what we're seeing is a direct response of the platelets that are in flow to the exposure of the subendothelial collagen that is now exposed as a result of the injury. Now, they are recruited to the injury sites, they become activated, they secrete their cargoes, they adhere to the wound, and also they stick together. Now, this plug is not stable at all under flow. So it is followed by the formation of the secondary hemostatic plug. So more platelets are recruited to the site. There's more secretion going on, more activated platelet adhering to the wound, and then there is the generation of thrombin, which is a potent agonist that eventually leads to the formation of fibrin from fibrinogen and then forms that mesh that seals, pack all the cells together and seals and seals the wound until healing occurs. Now, platelet role in this is actually a lot more intricate than this. Now, we've known as far back as 1978 that platelet balloon during hemostasis in order to stop the bleed. We see this from the work of Wester, which I'm showing you here. So Wester showed in 1978 that both in the primary hemostatic plug and in the secondary hemostatic plug, that there is the formation of balloon platelets. So you see here, I'm trying to indicate in yellow area balloon platelets, as shown by Wester, and he even mentioned that there was fibrin deposition between this. So there is some morphological changes going on there, but this is how far we can see as far back as 1978. But since then, there have been advances in microscopy. And we now know a lot more about the ultrastructural changes that platelets undergo during thrombosis and hemostasis. So you see SE on the scanning electron microscope of the platelets here, spectrum adhering to collagen fiber, it becomes activated, it goes through changes, which I'll talk about in detail later on. And here we are looking at the total example of the platelet body and the balloon. Now, as striking as these images are, they actually still leave unanswered fundamental questions about the cell biology of thrombosis and hemostasis. So what are these questions? The first, obviously, is why do platelets undergo remodeling or procoagulant membrane ballooning? Why do they go through membrane ballooning? How do they do it? And can we monitor PMD analysis for innovation for clinical translation? But during the course of this talk, I will be referring to the platelet phosphatidylserine a lot. So for the purpose of trainees that are listening, this is a phospholipid, it's a component of the cell membrane. In the literature, it is said to be involved in necrotic signaling and other roles, but it does a lot more. Now, in the crescent platelet, the inner leaflet is made up of the phosphatidylserine. This is a negatively charged membrane. And once the platelet becomes activated, what it does is that it actually flips outwards this inner leaflet. So now we have a negatively charged membrane exposed on the platelet membrane. This actually, it's a catalytic site for the docking of coagulation factors that are already in the plasma and in flow. So now they can dock on the platelet. So we have activated factor 10, which is the convenience complex that we know is needed to drive thrombin generation. So what we're looking at here is that when this happens, the platelet essentially becomes a hotspot for thrombin generation. Now we can track when platelet becomes procoagulant in our experiment in vitro by looking at the binding, monitoring the binding of annexin 5, which is a protein that binds very strongly to phosphatidylserine. Then we tag annexin 5 with a fluorescent probe so we can see spatially where this is happening on the platelets. Now, in the grand scheme of things, platelet regulation of hemostasis and thrombosis is dependent on three functions of the platelet. First is the ability of the platelets to secrete from its alpha granules, a procoagulant agonist, and the dense granules also agonists. Then we also have the ability of the platelet to aggregate. We saw that in the formation of the hemostatic plug, primary and secondary. This is mostly driven by integrin alpha 2b beta 3. But then there is this important aspect of their function, which we call procoagulant membrane dynamics, or PMD for short. So I will use this acronym a lot during the course of this talk. You'll be hearing PMD. It means procoagulant membrane dynamics. And it's really referring to the whole changes, the composite changes that platelets undergo in order to amplify thrombin generation. Now, a big part of this is the inflation of the platelet membrane into a balloon. But there are also other aspects of it. This is we have the membrane exposure of PS, then we have procoagulant spreading, which I'll talk about pretty soon. And then we have membrane, the breakdown of the membrane itself, like the circulation, there is expression of protein in the membrane, and there's the formation of functionally segregated phenotype. So all this is put together to systematically analyze, and that is called what we call the procoagulant membrane dynamics. Together with secretion and aggregation, they determine the rate and extent of localized coagulation. So what is PMD? This is a good point to start. And why does it occur? Now, to address that question, we look at the spatiotemporal dynamics of membrane ballooning and postcoital serine exposure. So here you are looking at a scanning electron microscope of a single platelet adhering to collagen fiber. The platelet becomes activated. The response is what is being captured in the video here, and also in the still images here that are taken from different frames of the video. So you will notice here that the platelet is actually capable of two kinds of deformation. So there's the one that I'm illustrating in magenta here. I call this blebbing. And then there is the deformation that I've highlighted. The one I've cyan blue is what I call blebbing. And the one in magenta is what I call ballooning, membrane ballooning. Now, we went on to characterize these two forms of deformation, membrane blebbing, ballooning, and look at the different phases of ballooning. And so we can talk about it in detail later on. But what is important for the purpose of this talk is that functionally, ballooning increases the surface area of the membrane for PS exposure. So if I can just draw your attention to frame 9 here, you will notice that at this point, both the platelet body and the platelet balloon itself have accumulated connectin 5, meaning that they are now able to allow for the docking of factor 5, factor 10, and they can become a hotspot for thrombin generation. So this way, the platelet is able to suddenly increase its surface area and amplify coagulation. You see this in detail as the talk goes on. Here you are looking at the platelet and SCM, the scanning electron microscope of the balloon platelets. We can see balloon platelets when we flow blood over whole blood now over collagen. So here I'm doing a Z section going up and down in Z section, and hopefully you can see as I try to highlight with the yellow arrows here, pointing to areas where there is balloon here. And then also in our model of in our in vivo model of thrombosis, we can also see balloon platelets, which we're looking at here. This is a carotid factor like what Western saw in 1978. So there is that evidence that platelet balloon, and there is there's solid evidence behind that. Now another observation that we made is that platelets are not born to be equal in their hemostatic response, especially after exposure to sub-endothelial collagen. Now under this condition, in that they can form at least 4 different phenotypes that are distinct, we believe are distinct in function. To be able to visualize this, what I'm doing here is I'm switching from bright field mode to fluorescence mode, and at the same time, I'm going up and down in Z section. So I hope you'll be able to catch this phenotype as I now try to describe them. So if you could follow me as I do that. So we have the non-balloon, non-spread. So at sections is non-balloon, and they're also non-spread, so at lower results. Then we have the BNS, the balloon non-spread. These are balloons, as you can see here, and they are not procoagulant spread on the lower section. Then we have the conventional spread non-balloon platelet. This is the kind of one we know this kind of spreading platelet very well. We call it the fried eggs. Then we have the balloon and procoagulant spread platelet. So if you notice here, it's really tricky to see this one because they appear to be ballooned at higher Z section, but when you go lower, you will see that these are actually procoagulant spread at the bottom. Now, PNS and procoagulant spread platelets are actually a distinct population. And again, as still images from that video, you will notice that they are the ones that have accumulated annexin 5 the most. So in other words, they are the ones providing the catalytic surface for the docking of coagulation factors. So we believe this phenotype among platelets will be driving thrombin generation. So perhaps we might be able to in future selectively target these platelets for antithrombotic effect. And we might be able to also use this platelet phenotype So again, I'm showing you a 3D reconstruction of the image platelets. I just want to draw your attention to this side. So everything you can see in red here is actually one platelet, a single platelet. And that is a balloon. You can see the balloon structure, and then the procoagulant spread part of it that is surrounded by 1, 2, 3, 4, 5 conventional spread non-balloon platelets that we believe are contractile and are able to and are performing a distinct role. In fact, there is mounting evidence that there is division of labor among platelets during the hemostatic phase. I'm interested in understanding this phenotype of platelets. So we did a 4D imaging to look at what kind of spreading we are looking at. And if you just pay attention to what is happening here in this 4D video going on. So we notice here that the kind of spreading is different from the conventional filopodial, lamellipodial spreading. This is more or less a microparticulate spreading that we are looking at. That's what we called it. So you can also see here in the still images here. Another thing that we notice is that the procoagulant membrane eventually breaks off to form multiple microvesicles. So this might be the way of platelets to generate microvesicles. We notice also that procoagulant spreading temporarily correlated with ballooning. So in an experiment where we've been able to halt or restrict that ballooning, we decrease procoagulant spreading by 9-fold. So it is possible that ballooning is one way that platelets allow for the formation of microvesicles. So a big question to ask at this point is, are the balloon platelets, as we've seen them now, capable of supporting thrombin generation as we have seen? Well, the answer is yes. And this is because we were able to visualize the binding of recombinant factor 5 and factor 10, which is the protein complex on the platelet membrane. We were also able to use an antibody to throw for thrombin to be able to visualize thrombin on the membrane of the platelet, and that's what we're looking at here. Now, during this course of experiment, we also noticed, that's widely reported in the literature, that the procoagulant platelet is under oxidative stress. So perhaps this is another feature that could be used to identify and to selectively target this platelet phenotype for therapeutic effects. Now, another observation that we made is that as thrombin is being generated, it can actually lead to synchronized calcium signaling and synchronized membrane balloon, and also mediate a burst in microvesiculation. So here you are looking at plasma platelets, human plasma platelets that we are allowed to adhere to collagen. Now, somewhere in the middle here, I took off the dye here, but somewhere in the middle here, there is the procoagulant platelets are there. So they are generating thrombin that is diffusing outwards. And what we see here is that now causing the calcium signal to go from asynchrony into synchrony. And that is associated with synchronized ballooning and then the burst in microvesicle from the aggregates to the surrounding. Now, so this is what I'm trying to highlight here. And the rest of the experiment here is really where we used to determine that the effect we are seeing is an effect due to thrombin. So by way of summary of what we talked about to this point, what and why is platelets PMD? Now, we know that platelets increase surface area for binding of clotting factor 5 and factor 10. They can increase the procoagulant response to membrane ballooning, microvesiculation, and the amplification of thrombin generation. This brings us to the question of the mechanism. What mechanism is driving platelet procoagulant remodeling? Big question. We did a lot of experiments, again, using our imaging approach. And what we find out that when we look at membrane blebbing, membrane ballooning, procoagulant spreading in the absence or presence of chloride ion, we notice here that chloride ion is very important. First of all, the chloride indicator that we use in this experiment is one that detects chloride entry by collisional quenching. So when you see a decline in a loss of fluorescence, is actually corresponding to chloride influx. And what we notice from the collation of the data is that we are able to significantly attenuate ballooning but not blebbing in chloride-free medium. And when we also block chloride by the use of chloride channel blocker, in this case calcium activated chloride channel blocker, we also block membrane ballooning but not blebbing. Similarly, when we remove sodium from the medium, Then we also noticed too that we can attenuate or significantly block ballooning. That's what we're looking at here, but not blooding. Then when we analyze the composition of different phenotypes, we notice that the balloon and procoagulant spread platelet that we talked about earlier on as being the one, mostly accumulating connectin 5 and like to be driving thrombin generation. The formation of those phenotypes is necessary for modeling in platelet is water entry. And we demonstrated this in experiments where we challenged platelets with hyperosmolar conditions. Immediately, we noticed that again, under this condition, the formation of balloon and procoagulant spread platelet was significantly attenuated. Then we asked the question of the channel for the water entry. So what we did using alpha-41 knockout mice, we established that water channel alpha-41 is actually required for water entry. But if you notice here, this is the wild type. We see the procoagulant spread platelets here, but if we compare that with the knockout here, what we are seeing really is more conventional spread non-balloon platelets. Formation of microvesicles is analysis, and we saw a significant decrease in microvesicillation in the alpha-41 knockout mice. Also using this model, we were able to demonstrate that knockouts of alpha-41 ablation will lead to suppression of thrombosis in our in vivo model of thrombosis. So here you're looking at platelet levels. Then this is the wild type. You see, I can just draw your attention to frame 4 here. You see the thrombos that are built up here and that have been significantly attenuated in the nature. Now, all this is happening, but when we look at hemostasis, that doesn't seem to be affected. So tail bleeding seems fine between both the wild type and the knockout. Now, this is interesting because there are not really many targets out there, molecular targets out there that are able to distinguish between thrombosis and hemostasis. So we are doing more work in this area. Mechanism that is associated with platelet procoagulant modeling is the disruption of the microtubule. Now, if you think about it, this is due to the increase in internal hydrostatic pressure. So we have sodium coming in, with chloride coming in, and osmotically obliged water is also coming in. So all that is increasing the internal hydrostatic pressure within the platelet and pushing against the rigid microtubule to the point where it starts unwinding. I hope you can see that here. I try to highlight it in the green arrow here. So the unwinding of the microtubule. If that is gone, then the platelet is then able to deform as we see it after. So to summarize this part of the talk in terms of mechanism, we know that platelet PMD results from the physical disruption of the membrane cytoskeleton interaction. On the subject of the molecular mediators of platelet PMD, is it only just alpha-41 as we earlier on? We know that for water to go into the platelet during that process, alpha-41 plays a key role. We've gone ahead to examine other potential targets, molecular targets. We've seen that for salt entry, calcium activated platelet channel is important. Recently, in some of our patients, we've been able to establish that CLIP1 and CLIP4 might also be needed for modeling, for procoagulant modeling. Then we think that non-selective cation channel will be playing a role, and that there is a role for carbonic anhydrase enzymes that more recently we've been able to demonstrate that facilitated glucose transporters GLUT1 and GLUT3 are also playing a role in the remodeling. So closely at the molecular targets carbonic anhydrase 1 and water channel 1, alpha-1, The interesting thing about this target is that when you inhibit them or block them, plate them, what happens is that they can regulate platelet PMD in vitro, in vivo, they block thrombosis, but they don't affect granular secretion. Significant. If you think about it, we have the current antiplatelets clinical use. They target P2Y12 like clopidogrel or aspirin targeting FOX1, and they ultimately block secretion. So the release of ADP, the release of thrombosin A2. So, but they needed to do that in order to do their job. But what they are also doing is that by blocking secretion, that we are blocking the good things that come out of platelets. So the content of cargo, platelet cargo that are needed for genesis and for maintenance of vascular integrity. And sometimes clinicians have to combine two antiplatelets for efficacy, as in during percutaneous coronary intervention, that again leads to significant bleeding. So to be able to identify a secretion-independent target for antiplatelet effect is really good, because then we can control blood clotting without significant bleeding, without compromising vessel integrity. And I think it brings us a step closer to the precision medicine thrombotic therapy. In terms of the signaling pathway for the formation of procoagulant platelets, well, in our hands, this has been predominantly driven by GP6. So this pathway here is where we see a lot of action going on. So P3 and release of calcium from the start. But we can also have a ballooning, a procoagulant remodeling, some form, well, when we target the G protein coverage. So can we apply the study of platelet PMD in the clinical setting? But the answer is yes. For example, Dr. Vivian in the University of Sydney in Australia, she's designing probes and doing clinical tests to recognize and target procoagulant platelet. Then Dr. Paul Armstrong in London is studying balloon platelet in the peripheral blood of trauma patients. He's trying to link that with excessive bleeding and mortality. Here in Calgary, we are looking at platelets role in the pathogenesis of progression. Here, we are looking at patients with antiphospholipid syndrome, hypertensive complications. So pregnancy, gestational hypertension. We are doing this in severe infection, in metastatic bone disease, and inherited bleeding disorder. And in all of these patient flows, what we see is that systematic analysis of PMD is actually to understand the events that's going on and the mechanism in this cohort. So in terms of innovation, analysis of PMD is also helping us to identify new targets for new drugs. Also using the analysis of PMD to develop a novel device for procoagulation analysis. Now, the whole idea is to be able to predict the mistakes in the status response period. I'm going to share highlights of some of these studies with you. The first one I want to talk about is our findings in antiphospholipid syndrome patients during pregnancy. Now, with the help of Lupus Canada and also a team of clinicians, we are able to assess the peripheral bloods of patients during pregnancy. So why this study? Well, antiphospholipid syndrome is an autoimmune condition and potent hypercoagulated state defined by thrombosis, are known to play a role in thrombotic APS. But the mechanism of platelet procoagulation that is operating in patients with APS to be able to identify novel targets, potentially improve both thrombotic and obstetric outcomes in these patients. So what we did was we began by looking at platelet PMD in antiphospholipid syndrome patients during pregnancy. So we followed the pregnancy of 2 pregnant APS patients. And for our control cohort, we also followed the pregnancy of 2 healthy pregnant control participants. plasma platelets of the healthy pregnant control that we've allowed to adhere to an inert surface. We put that surface is serum modeling. We don't expect any significant activation. So whatever we're seeing here is what we use. Back to platelet for alpha 2 beta 1, just as a platelet marker, this is a receptor on the platelet membrane. It tells us that we're actually looking at platelets. Then we looked at the exposure of P-selectin as a marker of platelet activation. Then we also tracked annexin 5. I talked about this earlier on, to use that as an indication when that the platelet has externalized phosphatidylserine. And in most of these experiments, we also use a thrombin antibody to be able to tell when thrombin is formed on the membrane of the platelets. Now, if you compare the top panel, which is from the healthy pregnant, to the lower panel, which is what we got from the APS patient, you will see immediately, comparing P-selectin, for example, that there is increased platelet activation. What you are also seeing that I try to highlight in the yellow arrow there is that the platelet also ballooned. There is increased procoagulation here, monitored by annexin 5. And here I try to zoom in on one of the balloon platelets. This is what it looks like. In XY, we see the thrombin there. If I draw your attention to the 3D reconstruction, the movie here, spatial distribution of thrombin, you can see is right on the membrane of the platelets of APS patients. Pass into this observation, what we notice is that there is, in terms of the balloon platelet, there is significantly more of this in our APS patients compared to the healthy pregnant. And we also noticed that balloon platelets were present in all 3 trimesters of pregnancy of APS patients, despite full dose of low-dose molecular weight heparin and aspirin. So expressions, procoagulation, PS externalization, and thrombin, it followed the same pattern. It followed the same pattern. Now, so we know now that there is platelet activation and membrane ballooning in nucleus. But could this be due to some intrinsic factor of the platelets or due to some procoagulant agonist that is within the plasma of the APS patient? Well, to address that, we did some mixing experiment. So what we did was we took the platelets from our healthy control and then essentially exposed them to plasma from our APS patient. I immediately saw evidence of platelet activation and membrane procoagulation. Just show you some of the images, the PMD images that we got. So here on the top panel, we are looking at the healthy pregnant control, again, marked for alpha 2 beta 1. So we know that these are platelets. And then for P-selectin, we see some activation there, and then annexin 5, one balloon platelet there, and then thrombin. But if you compare that, the same healthy pregnant control platelet that have now been exposed to APS plasma, you see significant increase in platelet activation. Then there is also increase in the number of balloon platelets. Then there's also increase in intensity of annexin 5. Thrombin on the membrane. So clearly there's something going on there, showing that APS plasma can. From this, we see that this stimulatory effect can go up to about 20 of week 28. For this, we don't know yet, but there must be some procoagulant agonists in the APS plasma. So how is platelet activation and membrane procoagulation being driven in APS patients? So the sort of changes that we've seen in this platelet is something that brings back memory. We know that the transformation of platelet into procoagulant phenotype balloon platelet is big pathway we talked about earlier on. So that was our first trial. So we tried to look at the possibilities that SIC and PLC-gamma2 mediate platelet procoagulation and membrane platelet activation and procoagulation in APS platelets. And here you see staining for phosphosig. You see phosphosig is increased in APS but we're able to attenuate this in the presence by pre-incubating with a sick inhibitor. Similarly, we went back to our sick experiment. We exposed platelets of healthy pregnant control with plasma from APS. Immediately, we see an increase in phospho-sick, which we're able to attenuate in the presence of a sick inhibitor. And here is the Western blot data showing the increase in phospho-sick in our APS. So there is platelet activation. It's occurring via CYP and PLC2. But the big question is, can CYP inhibition control platelet procoagulation in APS? Right, I would say. Because when you look at the data closely, stimulated with APS plasma, we see that CYP inhibition is able to control in this one's ballooning, P-selectin expression, and membrane procoagulation. However, when we focus on APS platelets themselves, we see that CYP inhibition is unable to rescue APS platelets from ballooning, P-selectin expression. So this really is pointing to the fact that the APS patients have APS platelets are pre-activated in circulation. Summary of our findings here. We know that platelet activation in ballooning APS, we know that there is some procoagulant agonist in the plasma. Ballooning that we're seeing there is occurring via SYK and PLC2. And we also now know that SYK inhibition procoagulation APS during pregnancy. So moving forward, we would like to look at the role of complement activation in this procoagulant state work. Pregnant patient this time around with thrombotic APS and compare with controls and other aims that we achieve. Now, the next story I would like to share with you, again, where we try to demonstrate the translational powers of PMD analysis is in preeclampsia. Preeclampsia is a hypertensive complication of pregnancy. Not good for mother, not good for baby. 18 million pregnancies every year. So it is defined as the onset of hypertension at about 20 weeks or so of pregnancy, as associated with proteinuria, and is distinguished from gestational hypertension by. Also know that platelet activation is involved in the pathophysiology of preeclampsia and contributes to the proteolytic state of the cell. Now, what we don't know is what mechanism starts it, what initiates it, and what sustains the platelet activation in preeclampsia. We also do not know how platelets may be involved in the pathogenesis of the hemostatic abnormalities in preeclampsia, because we know that these women bleed more. So to address this, we looked at platelet procoagulant membrane dynamics using our imaging approach. We examined the peripheral blood from preeclampsia, the preeclamptic women, and compared that with healthy pregnant gestational hypertension, and we also followed the non-pregnant female controls. Now, what was immediately obvious to us is that preeclampsia platelets are pro-aggregatory, and there is really no specific pattern here because we see them come in homotypic or heterotypic aggregates. But what is clear here is that we see more of these microaggregates in the plasma of our preeclamptic patients. And then we have gestational hypertension, somewhat following that trend, was significantly different from healthy pregnancy. When we analyze platelet PMD in this cohort, pay attention to the lower panel here with preeclampsia cohort, we see I'm pointing to a microaggregate that we form here, increase P-selectin, increase NSN5, increase membrane signal, all pointing to a pre-activated and procoagulant phenotype. And we here they confirm our observation that there is significant platelet activation, coagulation, and formation of thrombin on the membrane of the platelet in preeclampsia compared to without pregnancy. So this brings us to the big question, which is really always how does it happen? Pregnancy sustains platelet activation, pre-activation, and coagulation in preeclampsia. Well, to cut a long story short, we think that this is driven by how platelets in preeclampsia handle glucose. Glucose capacity and higher glucose affinity than GLUT1. Let's put that in mind. Same platelet, GLUT1 is usually on the membrane. And we think its role here is to allow for some glucose coming in to maintain basal metabolic activity by facilitating digestion. However, when there is stimulation, now again, we do not know what that stimulant is or how it's happening. But when there is stimulation, that this then allow for the alpha granules that are carrying both GLUT1 and GLUT2 and are proximal to the membrane to move towards the membrane, fuse with the membrane, lead to the expression of GLUT3 on the membrane, and then that results in the influx of glucose into the platelet. Now, the influx of glucose is now providing more energy for the platelet, which then allow the platelets to repeat the same thing, but with more intensity by going deeper into the cytosol to recruit granules, allow them to fuse, and then shuffle them to the membrane, leads to the expression of GLUT3 on the membrane of the platelet. And of course, GLUT3 doing what it knows how to do best then allows for influx of glucose into the platelets. Now, under this condition, there is enough energy for the platelets to continue to degranulate, and there's also enough energy for the externalization of phosphatidylserine happening. So this is why the platelets are pre-activated while they are in circulation. Now, when you expose these platelets to collagen, for example, during cesarean section, you will see here that the response of the preeclamptic platelets is significantly diminished. And we think this is contributing to the increased bleeding that we see in preeclampsia patients. We are also using PMD analysis to dissect inherited bleeding disorders in collaboration with clinicians here at Fortis Hospital. We're looking at hemophilia A, hemophilia B, and recently we looked at the Montreal platelet syndrome patients, and we immediately were able to use PMD to breakthrough bleeding that clinicians are seeing. And for example, we look at panel C here compared with panel F, and we notice that when exposed to collagen, the platelets of the MPS platelet are not able to balloon. You can see a minimal coagulation happening, and it's in fact binding the regular as compared to the top panel where it's healthy, controlled, strong ballooning, strong coagulation happening, and the numbers also show that there is decreased coagulation in response to collagen. So this might explain the breakthrough bleeding that clinicians are experiencing. Another thing that we're using PMD for is for innovation for precision medicine. Here we are trying to advance the systematic analysis of PMD into a point of care system, able to monitor hemostasis and predict the tendency to bleed or to clot. For example, one area where we are using this, we keep expanding on this, is we are looking at platelet PMD before, during, and after cardiopulmonary bypass surgery. And what is happening is that the analysis is able to, the PMD fingerprint is able to categorize patients into distinct phenotypes. And this phenotype, in turn, we see are correlating with the total amount of blood loss 24 hours after surgery. And the first software that we try to put together for the systematic analysis of PMD to do this correlations. So on that point, we'd like to conclude. So in terms of the mechanistic insights, PMD, procoagulant membrane dynamics, we know that platelets here to vessel matrices and they undergo unique transformations that are driven by salt and water entry. In terms of the clinical translation from PMD study, we also know that elements of PMD are critical amplifiers, regulators, and sensors for thrombosis and hemostasis. And for innovation, we are now beginning to see that when we systematically analyze platelet PMD, it can be able to predict bleeding or clotting tendencies. I also want to say thank you to Bata Health Services, the Human School of Medicine, for their support, the Canadian Hemophilia Society, and the British Heart Foundation for support. And of course, to all my amazing collaborators.
Great, thank you, Jaffe, for a fascinating seminar. If anybody has any questions, please submit them via Q&A. And we have two questions coming up here from Maha Othman. Comment, fascinating work. Would love if you can kindly explain a bit about your basic methods, microscopy type, and how these platelet images are acquired.
I would love to. Well, we use the confocal system, and we find out that from magnification of about 60 to about 100 or 120, we start getting good data. We also try to acquire at night press with good resolution. The problem usually is could be noise. So you have to watch out for signal to noise ratio. Description of our method. Also, we in the JTS paper of 2023, we also described that, but with data that we've gotten from confocal microscope. We also do use dark field microscope, and then we have dedicated softwares that we use for analyzing the images. Like I said, you have to really deal with noise, so we develop point spread functions that we then filter the images through to be able to take away all stray light, and that allows you to get very sharp images. So we have softwares that we use for that. But I can provide you with more information if you reach out to me via email.
Great, thank you. There's a follow-up question, Jaffe. It's you mentioned a translational value in metastatic breast cancer. Your sound was cutting off, so could you please elaborate on how MDP valuable and where is the gap in knowledge?
Well, that work is ongoing. What we've seen is that PMD can actually help to stratify the patients. That's one of the key findings there so far. So even before surgery, this patient with metastatic bone disease that are going through surgery, they are so different. So some of them are already pro-aggregatory, and some of them are not. So it's almost like PMD is saying that they needed to be treated very differently. This is before surgery. So they needed to be observed very differently. Some of them need to be on antiplatelets straight away. So it's an ongoing work, but that's what I was trying to explain there. I didn't present any of the data there, but that's it's summary of the potential. So we can use PMD to stratify this patient and treat them differently. So we are essentially having a method that we can use to individualize the way we treat our patients.
Okay, great. I've got a couple of questions actually, Jaffe. So in terms of, you mentioned, in your talk that platelets are not born equal. Do all the platelets have the ability then to generate these, membrane balloons, procoagulant membrane balloons, or do certain subpopulation of platelets have, let's say, increased aquaporin, or any of the other, GP6 or any other the proteins that are thought to be involved, and can you look at sort of potential, if so, ratios of some of these platelets that might have higher levels of some of these proteins in various pathological states, I guess?
Well, it seems that they are pre-committed. It almost seems that they are pre-committed. And one of the things we are still trying to understand in the field is what pre-commits a platelet to either stay as a CSMB or to go into BAF. We don't really know what does that. It's something that we're working on, myself and Vivian, it's something we are looking into, because if we can recognize this platelet in circulation, then that opens the way for selective targeting. Again, there have been studies looking at the transcriptome to see is that what determines it. Do the procoagulant platelet have a unique transcriptome as compared to the conventional spread balloon? Well, the result is not definitive yet. So it's work ongoing. I wish I could answer your question better on this, Paul.
Okay, great. And so my second follow-up question to that is along the same lines, because my lab also studies platelet heterogeneity, and I get this question asked a lot of me very often, and it goes back to the megakaryocytes. And so are there megakaryocytes that sort of preferentially generate platelets that are more procoagulant and form these balloons, or not? Have you looked at that at all?
No, I haven't looked at that. We haven't looked at that, but it seems like it's either they are loaded with different transcriptome to begin with, or there is some change that happens while in circulation. Another hypothesis could be that the older platelets are the ones that become procoagulant, that tend to go to BNS or procoagulant, the BAFs, the procoagulant and balloon and procoagulant spread. Because if you look at it from the perspective that the body is dumping about 1 million platelets per second into the circulation, then, and in terms of how long they stay before they are cleared, you have a huge dynamic range in age. So some people believe that perhaps the older platelets are more likely to become procoagulant upon stimulation. But again, this is all theories that we are working on.
Great, thanks. Okay, I'm not seeing any other questions coming through the Q&A. So just want to thank you again for the great seminar. And the seminar recordings are also available online on the CanVector website. Thank you very much.